Hello everyone, today in this video we will solve some conceptual question on genetics for quick revision. Okay, so let's start with the first question. Dihybrid cross proves the law of segregation, dominance, purity of gametes, or independent assortment. As we know, during gamete formation, the unit factor of a pair segregate randomly, and so gametes are pure for a particular trait. If we take example of capital T and small t, then during gamete formation, this unit pair, okay, of a gene of a particular trait will segregate randomly and goes into different gametes. So the gamete will be pure for a particular trait. Okay. Likewise, for the trait of the color of flower, which we take as capital W and small w, will separate randomly and independent of the separation of different trait. Okay, different trait, and this proves the law of independent assortment. Okay, so dihybrid cross proves the law of independent assortment. Okay, so the answer is option D. Since independent assortment itself includes segregation and purity of gametes, so the best answer among these will be independent assortment, and dominance has no role in this. Okay, it's a phenotype. Question number 2 says, how many types and in what ratio the gametes are produced by a dihybrid heterozygous? Options are 4 types in the ratio of 9:3:3:1, 2 types in the ratio of 3:1, 3 types in the ratio of 1:2:1, or in 4 types in the ratio of 1:1:1:1. Okay, as we have seen in the previous conclusion that gametes form are pure for a particular trait, so the ratio will be 1:1:1:1, since each gamete receives a unit factor of a particular trait. Okay, and to calculate number of types of gametes we use 2 to the power n, where n is the number of hybrid or the heterozygous trait. For dihybrid we have 2. Okay, for example capital T small t, capital R small r. Okay, so we have 2 heterozygous condition. So if we put 2 in place of n, we will have 4. So the gametes form will be of 4 types. So the correct option is option D. Okay, moving to question number 3. Who rediscovered the result of Mendel's experiment? Options are D. Varese, Shimark or Correns, D. Varese, Shimark, Morgan, Shimark, Morgan, Correns, and none of the above. The correct answer is option A, D. Varese, Shimark. Question number 4 says, due to the cross between these two, the resultant progeny showed how many percentage plants would be tall red flowered. Okay, to solve such question, we will take the different traits individually. So for this, we take the tallness independently and the color of flower independently. Okay, so we can see after the cross we will get the tall plants. Every plant will be tall. Okay, and if we take the color of flower in case and draw its Punnett square which is be like this. Okay, so the result will be capital R small r small r small r. Okay, so here we can see half of the flower color are red and half are white. We are taking white in this case. Okay, so here you can see half are red and every plant are tall. So The correct answer will be option A, which is 50%. Question number 5 says, when two different genotypes produces the same phenotype due to environmental difference, then each one is known as option A, phenotype, option B, phenocopy, option C, progeny, or option D, none. The answer for this question is option B, which is phenocopy. Okay, for this question, let me know in the comment section, is it appropriate to call it a case of convergent evolution? Okay, think on it. Question 6 says, probability of forced son to a couple is. Okay, here probability means chances of having son in every meeting. As we know, there is a 50-50 chances of both boy and girl progeny, or 25% in each case, according to Punnett square. Okay, so for first mating, probability of having a son will be 2 out of 4. Okay, if we draw the Punnett square, we draw it xy, xx, then here you can see 2 females and 2 males. So probability of having son is 2 by 4 in first mating. Okay, so according to Punnett square, for 4 son we have 2 by 4 into 2 by 4 into 2 by 4 and 2 by 4 times, which will gives 1 by 16. Okay, so the correct option is option C, 1 by 16. You can pause the video in between to solve such questions by your own. Okay, now moving to question number 7. ABO blood group is an example of option A, epistasis, option B, multiple allelism, option C, pleiotropism, and option D, complementary genes. Okay. As we know, ABO blood group is an example of multiple allelism. It occurs due to mutation. And here, pleiotropism means a gene which controls more than one character. Okay. And complementary genes, the two pair of non-allelic genes essential for expressing a particular trait in a dominant form. Okay, and we left with the epistasis, which means when a gene prevents the expression of another non-allelic gene. So here correct answer is option B, which is multiple allelism. Okay, now question number 8, which says allele A1 has nil abundance in population of A1 and A2. These two alleles are present in a population, then abundance of A2 is. Since we know about the Hardy-Weinberg equation, which says P plus Q is 1. Here P and Q are A1 and A2. According to question, A1 has nil abundance, so A1 will be 0, so A2 comes out to be 1. Okay, so the answer is option C. Question number 9 says, the migration of a gene into a population from another population by interbreeding is called gene pool, gene flow, gene drift, or gene erosion. First, we should know about the terms in options. Okay, so gene pool
मस्त जोक मारा रे ये हंस रे हलकट हंस
Gene pool refers to the stock of different genes in an interbreeding population. Okay, gene flow, transfer of genetic variation from one population to another. Genetic drift, It is a change in frequency of an allele within a population over time, and this change in frequency of alleles must occur. It is a change in the frequency of allele within a population over time, and this change in the frequency of allele must occur randomly in order for genetic drift to occur. Okay, and gene erosion, also known as gene depletion, in which limited gene pool of an endangered species diminishes. Okay, so the answer must be option B, which is gene flow. Now moving to question number 10. Frequency of recessive allele for a certain trait is 0.20. What percentage of individual in the next generation would show dominant trait? Okay, to solve such questions, you need to read it carefully, as in this, as it didn't say the frequency of pure dominant. Okay, instead only dominant is given, so it will include the 2pq as well. Now according to Hardy-Weinberg, we have p plus q is 1 and p square plus 2pq plus q square is 1. Okay, here 0.20 is the value of q, recessive trait. Okay, so we can find the value of p, which comes out to be 0.80 from this. Okay, so now if we put it in 2pq, we will have 2 into 0.8 into 0.20, which will be 0.4, 4 8s are 32, 0.32. Okay, and p square will give, p square is 0.8 into 0.8, which will be 0.64. Okay, and now we need a dominant trait in a population, so by adding p square and 2pq, we will get 0.32 plus 0.64, which will be 0.96, and in percentage 96%. Okay, so the correct option is option D, 96%. Moving to question number 11, which says the existence within a population of a non-beneficial alleles in heterozygous genotype is gene flow, gene load, gene drift, and none of the above. As the name itself suggests, gene load. Okay, the answer is option B. And now we have completed with the part 1 of genetics. Do like and subscribe the channel for more such videos. We'll meet again in another part of the topic. Keep learning.